Good morning, biology class. Welcome to April 23rd, Thursday. We're going to go through today's lesson quickly. So obviously step one is watch this lecture. Step two will be to practice some mutations. We'll do the practice after the lecture. Here comes the lecture. All right, so by the end of class, you guys should define and identify frameshift mutations. So remember, we're talking all about mutations. Mutations is when DNA changes. Remember, if DNA changes, then eventually the proteins are different too. All right, so we're talking about frameshift mutation. Now, a frameshift mutation is a mutation that changes the way the entire gene is read. So imagine that if this is the gene down here at the bottom, that there is a mutation, then all of a sudden every base after it is read incorrectly. To elaborate on this definition, we're going to go over two kinds of frameshift mutations. One, insertion. You see over here where a gene is inserted, and then every base thereafter shifts over to the right. In this example, deletion, a base is deleted from the sequence. And again, you see that this letter T that was right here gets deleted or removed from the sequence, and then every base shifts to the left. So let's slow down and go over each of those with a little bit more detail. All right, let's talk insertion. So let's imagine we had a sentence that was, the fat cat sat. Now, insertion would occur if a new nitrogen base was inserted into the sequence. Now notice, all the letters are still present, but now the frame of which they are read has been shifted. So instead of the RNA reading the fat cat sat and creating the appropriate amino acids, now it says ta, effa, tka, tsa, t. So again, this sentence is an example of insertion. Here's another. Imagine we had a strand of DNA, ATG, CAT, GTA, and an A was inserted, making all the letters shift. Notice that the G just shifted over. The CAT just shifted over one. The GTA just shifted over one. And now the codons being read instead of ATA are mutations, ATG. Instead of CAT, it's GCA. All right. And now remember, the whole idea of insertion is that it causes the RNA to transcribe incorrectly. And then the amino acid is different. That's the whole big deal. So if you remember how the codon wheel works, then you'll see that if there is an original DNA of CCC, AAA, TTT, GGG, and then it gets transcribed and then eventually translated, we're going to notice here the bases will shift. So watch, I'm going to shift the bases. I've inserted an A. Everything is shifted over one. The DNA bases have been shifted. And now, fine, one of the amino acids is still the same, but all the other amino acids have changed. So this might be, the amino acid for a protein, or this might be the protein for, heart muscles. But now the protein is mutated, and maybe it'll be, a somehow damaged heart muscle. All right, so the next one is deletion. So a deletion is when a base is deleted from the sequence. Here's that sentence again, the fat cat sat. Now imagine we deleted the letter H, tef, etk, ets, at. Now this sentence is read incorrectly. All right, again, here's the original sentence. We deleted a letter, the letter H in this example, and now the sentence is read incorrectly. Here's another example. In this case, we're going to delete the T. That's the second nitrogen base in this codon. So instead of the original strand, ATG, CAT, GTA, we delete the T, and now it's AGC. Notice how they've shifted over into a different codon. ATG, they've shifted over into a different codon. And then you have TA, and who knows what DNA comes after that. All right, so study this. And again, of course, the problem here is that as a result of mutation, the RNA transcribes incorrectly, and then the amino acid is different. And if the amino acids are different, the proteins are different. So here is our original strand. We are going to delete this C. These bases are all going to shift over. The mutated strand has that C deleted. So instead of GCG, it's GCA. Instead of AGT, there's that A, and that's GTT. And again, I'm not going to read them all, but notice how they've all shifted over. And again, the big deal here is that as a result of a mutated DNA, the transcription is different and the translation into the amino acid is different. So mutations cause change. You've just reviewed insertion and deletion as frame shift mutations. So let's take a little bit of a look at the practice that you're going to do. So for practice, you guys have to use the terms to answer these questions. Remember, it's so important that you explain how you know. So here are the vocabulary words. Here is your original DNA and your mutated DNA, and studying the original mutated DNA in problem 1. What kind of mutation is this? Is it frame shift? Is it insertion? Is it deletion? Is it 2 of these? Answer here and tell me how you know. Same deal. What kind of mutation is this? Then we ask you to write an example of frame shift due to insertion. So you'll write 3 nitrogen bases or 1 codon in each of these boxes, and then write a mutated DNA showing frame shift mutation or due to insertion. Same deal, except show frame shift due to deletion. And then here we want you to really understand what kind of mutation is shown and what is the effect of the mutation. In other words, is there a changed amino acid? There is a codon wheel on the last page for you to use. So here's the codon wheel. Of course, you could just Google search those too. All right, students, as always, reach out to Mr. Williams, myself, Ms. Check, Ms. Carr for any academic help. We hope to see all of you guys on this Google Classroom, and get ready for some changes. You guys have been doing all right, but next week we're going to make it even more user-friendly. Peace.